Chapter 1, Overview of Microbes and How They Shape Our History. So, the first thing about a microbe is the definition. So, textbook definition on page 4 of your textbook says, a microbe is commonly defined as a living organism that requires a microscope to be seen. And this is true. However, there are a few exceptions to this. For example, protists and some algae are large enough to be seen with the naked eye. But in general, all microbes need a microscope to be seen, hence why it's microbiology. Why do we care about microbes? Specifically, bacteria. We're interested in the enzymes that they produce. They have a really simple DNA, and that DNA codes for genes, and those genes make proteins. And for our purposes, these proteins are also called enzymes. And that is why we find them very interesting, because we're able to manipulate them to make what we want. We're also able to study them to see what it is about their specific genes that could make us very sick or very ill. But we use them in our everyday lives, and you're probably not even aware of it. There's many, many, many examples of how they're used. And why scientists like them and for the environment is that they're biodegradable, because they're from a living organism, and we use them in such as things like different foods, textiles like paints, laundry detergents, digestive aids like the Tums and Roll aids I gave you an example of in my notes. Also, in terms of textiles, think about fade-resistant paints that are now available for people that live in warmer places such as Florida. Traditionally, these homes were painted light pastel colors to help with the heat, but also part of it was that if you had a dark colored home like a maroon or a green or a brown home, that would fade in the sun over time. But now Sherwin-Williams has a huge plant in Central Florida, in Orlando, and they have devised a fade-resistant paint with the help of enzymes that are found in bacteria. So basically, there's so many examples of how they're used in our everyday lives. And I give you the examples of water from our oceans and lakes, which are taken up by mammals such as whales and fish, both of which are part of our food chain. In the soil, the microbes help to recycle chemical elements. And you're aware of this, especially in the fall in New England, when the leaves fall and you leave them there. And then you leave them through the winter, you haven't raked them up, you haven't blown them away, and then you lift up that leaf pile again in the spring, and it's all gross and slimy and whatnot. Well, the bacteria from our environment is helping to biodegrade those leaves to make us help with rich soil. Another example is photosynthesis. Microbes help with this as well, as some of their metabolisms depend on photosynthesis, such as algae. So just moving along, there's in our food, yogurt, cheese, wine, beer. Microbes help to produce all of these as well. Within us, they help with our digestion in our digestive tract and with the synthesis of vitamins B and K. And you might have heard of something, it's quite popular nowadays, probiotics or microbe implantation for people that have problems with their digestive system and their colon, where the bacteria has not been able to grow, or it's not and it's not able to produce the things that these people need naturally, so they will try to reintroduce that otherwise what's called normal flora back into people. That's why you don't want to mess with your intestines, your colon, your digestive system, because you have natural bacteria there, which is called normal flora, that really do help with digestion and synthesis of two important vitamins, vitamin B and vitamin K. These are helpful in blood clotting. So I had some terminology in my notes. The textbook has a lot of terminology. One of them is chemotherapy, the chemical treatment of disease. Bacteria help with this. So the first person, Paul Ehrlich, created arsenic-containing drug called salvarsan, and it was used to treat syphilis. Way back when, people used to die of syphilis, a sexually transmitted disease. Now they don't. Antibiotics are naturally produced by bacteria and fungi. People forget that fungi also produce antibiotics, and we all know about penicillin. We've all heard of penicillin. That was produced by a fungus, not a bacteria. And it was Alexander Fleming, by pure accident, that discovered the mold, and another word for mold is fungus, penicillium, that inhibited the growth of a bacterial culture. So it's kind of strange when you think about it that bacteria and fungi both produce antibiotics, but those antibiotics work against other harmful bacteria or fungi that we don't want in us. Because remember, this micro course is for allied health, and we're thinking of this in more an applicable clinical setting. So drug-resistant microbes, these are not our friends. These are the microbes that change shape and form so that the drugs that we develop are no longer useful. And I gave you the example for the ever-changing flu shot. Now that flu shot changes every year because there's the virus itself that causes the flu, or the bacteria that can also cause pneumonia. That changes from year to year. The vaccination for, in terms of if it's a virus, is made against the most popular virus from the prior year. The Center for Disease Control develops this and is in charge of what flu vaccination somebody will get from year to year. And it's different from year to year because these microbes are able to change themselves. They don't want to die, so they will do what they need to do to survive. So sometimes bacteria that was normally not harmful to people can become harmful in the long run. And we'll talk more about that. Modern developments in microbiology over time. Out of microbiology came bacteriology, mycology, parasitology, genomics, immunology, virology, recombinant DNA technology. Recombinant DNA technology, I just want to say, recombining DNA. You do this in a lab. And just as I said briefly to you in your notes, it says DNA is inserted into bacteria and other microbes, but generally we use bacteria because it has such a simple genome, and it can be used to make large quantities of a desired protein. So you mix the DNA together, put it into a bacteria, and you make the bacteria produce it again and again and again and again. So you get large quantities of a desired protein. Microbes in human welfare? Well, not only do some of them harm us, but they help us a lot. So in terms of sewage treatment and bioremediation, we are very thankful for bacteria because they decompose organic matter in sewage. And then we use bacteria to clean up toxic wastes. Hazmat teams, think about that. Deer Island, waste treatment plants. We use them in pest control. We use them in gene therapy, which is very, very popular area of research, where a virus can carry replacements for defective or missing genes into a human cell that otherwise we didn't know how to replace those missing genes, but a virus will happily do it. Now, human disease is what we're really interested in. So your normal microbiota or normal flora are, by definition, the microbes that we all have in and on our body that we live with every single day. By definition, the term resistance determines whether or not a person will contract the disease. So if you're highly resistant, it means you probably won't get the disease. If you have low resistance, because say you're an elderly person, say you're a newborn infant, and your immune system is not very strong, that means you have a higher chance of contracting the disease because you have low resistance. An infectious disease is one in which a pathogen invades a susceptible host. An emerging infectious disease, these are different. It's a new or changing disease showing an increase in incidence in the recent past or a potential in the near future. So this was a disease that you may have seen 200 years ago, then all of a sudden it's making a comeback. Or it's a disease that we thought was had been suppressed or eradicated, then all of a sudden cases start to pop up again. So it changed itself. Examples are the HIV virus, mad cow disease. How do you name these microorganisms? We're going to talk a lot about them, but it's important to know how to name them. So the genus is the first name that's capitalized and underlined, or you'll see it italicized. And then the specific epithet, or the species, is the second name that's not capitalized. And after a name is mentioned once, it can be abbreviated with the initial of the genus followed by the species, which is generally the way that you'll see it, especially in lab, like we deal with E. coli, B. subtilis, where its full name, the genus is Bacillus, and the specific epithet is subtilis. But once you name the bacteria once, its full name, the full genus written out, and the full specific epithet written out, the species, then you can abbreviate it. So classification of microbes, this was done not that long ago. For some of you, it seems like a long time ago, but for me, not that long ago, by Carl Voss in 1978. The classification was based upon the genetic relatedness of the organism. So the degree of relatedness is determined by comparing the total DNA sequence content for each organism, otherwise known as the genome. So microbiology, all these microorganisms, it depends how closely related their DNA looks to determine whether or not they are related, or their degree of relation. So prokaryotic taxonomic classification based on Bergey's Manual of Systematic Bacteriology. So this came out of that classification, what we call Bergey's Manual. And you'll have a chance to use Bergey's Manual in lab. Two other definitions that are very important: a strain and a bacterial species. So a strain is a group of bacteria derived from a single cell. So one little cell of bacteria then produces and replicates, and it's going to produce a bunch of other bacteria exactly like itself. A bacterial species are closely related strains. So bacterial species don't come from the same cell, but their strains are closely related because they have very much the same or closely related genomes. Their DNA looks quite the same. So classification of viruses. They are not placed in a kingdom at all because viruses are acellular, meaning they're not composed of cells and cannot grow without a host. The definition of a viral species, therefore, is a population of viruses with similar characteristics that occupies a particular ecological niche. So these, just like the bacteria, they're close. The key for species is closely related. Those two words, closely related if you're a species. If you're a strain, you come from the same cell. But it's just like having close relatives. Like your mother and father are the strain. The species are your brothers and sisters and your cousins. You're closely related. You have some of the same genetic makeup. Keep in mind that some microbes are prokaryotes and others are eukaryotes. Now, you should remember this from biology, from Bio 101. However, let me refresh your memory because you need to really know the difference between prokaryote and eukaryote. So prokaryotic characteristics, they have no nuclear membrane, meaning that nuclear membrane is an envelope that encloses the DNA. Their DNA is not associated with eukaryotic histones, or another name for these eukaryotic histones are chromosomal proteins. They lack membrane-enclosed organelles, and the cell wall contains complex polysaccharide called peptidoglycan. Now, eukaryotes are you and I. Think of that. You and I are eukaryotes. They have a DNA surrounded by a nuclear envelope. Our DNA is associated with histones. We have membrane-enclosed organelles. Our cell walls, if they're present, they're very, very simple. We divide by mitosis, and we're huge. We are much bigger cells. Think about Fenway Park being a eukaryotic cell and the baseball being a prokaryotic cell. That is an extreme difference in size. Also, I forgot to mention that prokaryotic cells, they divide by binary fission. They just simply split in half. Whereas we go through the whole phase of mitosis and meiosis for reproduction. Microorganisms and their cellular characteristics. Now, as painful as it may be, you need to know the characteristics that I have here for bacteria, for archaea, for fungi, for protozoa, for algae, and for viruses. This is what I'm holding you responsible for in terms of these specific characteristics. This is a sheet that you'd always go back to see, okay, what makes bacteria different from algae? What makes viruses different? These characteristics are the most important. Now, looking at the history of microbiology, you might really be scared because should I know these dates? Should I know the table that's in the textbook that when I'm reading chapter 1? No dates. I'm not going to hold you to any dates. And the tables look very, very intimidating in chapter 1, but they're just trying to give you an outline and introduce you to the history to say, so here is where microbiology played a role for a very, very, very long time, and they didn't really call the science microbiology then. Table 1.1 on page 7, 8, 9, you can see when certain diseases, very popular diseases, were discovered. So, but no names, no dates. You're just responsible for what I have on your sheet, like Robert Hooke, Antoine van Leeuwenhoek. What is spontaneous generation? What is biogenesis? What was Louis Pasteur? Look at all of the things that with this overachiever. Microbes can be present in non-living matter. Microbes can be destroyed by heat. Aseptic technique prevents contamination. Fermentation, pasteurization, vaccination, all of these things were Louis Pasteur discoveries. Then John Tyndall discovered the autoclave, the birth of the autoclave, because spores can develop, and then came the birth of the autoclave. The only way to get rid of bacterial spores is using high pressure and high heat, which is what we use to destroy all of the waste that you create in the lab. Then came Robert Koch, very, very important and used till this day, the scientific method of microbiology. He applied it to tuberculosis, and this is an example that's used over and over and over again in medical schools and graduate schools. So Koch's postulate, what it is, the simplest way to put this, it's a sequence of procedures which are used today to prove that a particular microorganism causes a particular disease. So 1, the microbe is found in all cases of the disease, but is absent from healthy individuals. Number 2, the microbe is isolated from the disease host and grown in a pure culture. We'll be making pure cultures when you perform street plate isolation. Number 3, when the microbe is introduced into a healthy, susceptible host, or an animal model, like such as a mouse or a rat, which are commonly used, the same disease occurs. These seem pretty intuitive, but way back when, this was not common knowledge. In number 4, the same strain of microbe is obtained from a newly diseased host. When cultured, the strain shows the same characteristics as before. So from this came the germ theory of disease. Microorganisms cause disease. And then, very important person, a woman in the history of microbiology, is Florence Nightingale and her notes on nursing. She was responsible for the first statistical analysis of infectious disease patterns. And we see this, how infectious disease increases in the summer. And because of this finding, it led to improved health policy for people. Because before that, during wars, more people died of infection than were actually killed in the war. They died because of wounds. Epidemiology came out of this. It's a chain of infection or transmission. And a type of epidemiology which is very interesting and applicable in healthcare is public health. It's the role of infectious disease in the health of large populations. And that's the job of the Center for Disease Control and Prevention. Boston University has a phenomenal school of public health. And there are lots of people that take epidemiology in terms of health epidemiology and follow the trends of, say, diabetes, high blood pressure in different populations of people all over the world, all over the United States, all over just Massachusetts, different areas. Edward Jenner and Pasteur, their claim to fame, vaccination and immunization. There are many ways to create vaccinations and immunize people. However, the goal is for the person to survive the vaccination, which is sometimes very difficult in creating them. We're going to talk about that in the last third of the course, vaccinations. However, it's not as simple as we would love to create a vaccination for every virus out there, but we don't want to give the people the disease. So there's many players that go into, that need to be thought out before you can actually give a vaccination to a person. Microbes help us live in our environment and adapt to changes. Although some microbes cause disease, some microbes are absolutely necessary for life on our earth and in our body to stay in homeostasis. Your body and our environment would not be in homeostasis without these microbes. And that's something important to really think about. Sometimes people want to disinfect their child and just put them in rubbing alcohol and constant bathing, 10 baths a day, and I'm exaggerating, of course. However, that's not good for a healthy immune system. You need your immune system to adapt and evolve with change and get used to its environment. And the way you strengthen an immune system is for it to be exposed to different environments. If you shelter a child and try and keep them safe and safe from disease and never get sick and whatnot, your child's immune system doesn't grow. So these microbes, they do help us, even though we're used to thinking of bacteria and viruses as very, very bad things that make us sick. And if we didn't have them, we'd be out of jobs in healthcare. So now, moving on to DNA. Now, this should be a review, but at minimum, you need to know what I have in my notes. So, by definition, genetics is the science of heredity. The genome is your genetic information within a cell. And we talked about eukaryotes and prokaryotes are a little bit different. Remember, eukaryotic genome has a nuclear envelope, whereas a prokaryotic genome, it's just inside the cell, but it's not enclosed in an envelope of any sort. Chromosomes are the way the cell's genome is organized. And remember, prokaryotes, their genome has no histones associated with it, but eukaryotes, they do. And that DNA is what physically carries the hereditary information. And then genes are segments of DNA found on the chromosome. So you start with the study is called genetics, that's the macro, and you're working your way down to the very specifics, the micro part, and that's genes. That's what we're interested in. So what do genes look like? So it's DNA within a cell that exists as long strands of nucleotides twisted together in pairs to form a double helix. You've all probably seen a piece of DNA, or you know what it looks like in your head. And what you need to remember, the DNA nucleotide pairs are A binds to T and C binds to G. Whereas RNA nucleotide pairs, the only difference is the U. A binds to U and C binds to G. And the structure of DNA explains its function. It's a linear sequence of base pairs that provides the actual information. So what matches up will provide the information. There's a complementary structure allows for the precise duplication of DNA during cell division. And these genes code for specific proteins. It doesn't matter what organism you are. All genes in humans or in bacteria code for specific proteins. And unlike anatomy and physiology, in micro we sometimes use the word protein and enzyme interchangeably because we're interested in what the proteins or / enzymes that bacteria produce. The genome is the genetic makeup, the characteristic of the organism specifically, a collection of genes that gives the potential for certain characteristics is the genome. Whereas the phenotype is the actual expressed properties, or what you can see, the collection of proteins that were made from the instructions of the genes. So for example, in humans, I have green eyes. My mom has blue eyes. My dad has brown eyes. So how did I get green eyes? Well, my mom and my dad both carry genes for green eyes, and therefore that's how I got them. So they had the genome for green eyes, but their phenotype was different, what was actually shown on the exterior. The bacterial chromosome is different, looks a little bit different than because it's a prokaryotic chromosome, than the eukaryotic chromosome. It's a circular double-stranded molecule of DNA that's attached to the plasma membrane. It's very, very simple. Whereas ours is a lattice, like a ladder. The bacterial chromosome is circular, but it's still double-stranded, and it's close to, in some textbooks will say attached to the plasma membrane. But remember, it has no envelope on the outside. So that's what makes it a prokaryote. One of the characteristics that makes it a prokaryote. And it's very, very simple, which is why we like to work with bacteria in the lab. So that's what I'm holding you responsible for in Chapter 1. I will get to Chapter 2 in the next lecture. Take care. Bye-bye. Thank you for listening to me.